Good morning, good afternoon, and good evening, everybody. We want to welcome you to our first webinar in a kind of two-part series that we're working on. This one is going to be focusing on streamlining your workflow, efficient bioanalytical sample preparation, and then in about a month's time, we will be focused on the LC troubleshooting portion. So my name is Rachel Lieberman. I am the the forensic and toxicology market manager here at Waters Corporation in our clinical business unit. I am here with Jonathan Denisau, who is a principal scientist at Milford, and he will be doing the presentation today. And I will introduce him here shortly. But while we wait for a few people to jump on, I would like to go over just a few housekeeping items and anything like that in case this is your first time using the ON24 platform. So here is kind of a debrief of generally what you guys will see on your console. And so we have a Q&A box. So please use that Q&A box to ask any questions throughout the webinar. We will have a Q&A session concluding the presentation where we will answer any questions that have been asked. In the center, you will see the slides for the presentation. And below those slides, you will find a resource list with any additional information and applications related to what we will be discussing today. On the right-hand side, you will see a speaker bio, as well as a survey. So please feel free to fill out this survey at any point during the presentation. We really like to hear all the feedback that we get from those that attend. So if you get a few minutes, please fill it out at the beginning or during the end. We are always looking for new topics for new webinars that we can provide to you guys that would be of interest. So if you have any ideas or things like that, please fill out the survey and let us know. Also, we will be conducting a few polls throughout this presentation to kind of gauge the audience, see kind of what background everybody has, and some questions of where we can tailor the presentation towards. Now, if at any point you are unable to see any of these windows that I've talked about, please refer to those icons in the gray at the bottom. And if you click on them, it'll allow those to expand at the top. So what I'd like to do next is to introduce our speaker. So Dr. Jonathan Denisau is a PhD as a principal scientist at Waters, received his bachelor's in biology from Allegheny College and a PhD in neuroscience from the University of Utah. He came to Waters back in 2011 after spending several years in the forensic toxicology and sports doping fields. And ever since joining Waters, he has focused on sample preparation and LC-MS solutions for forensics, clinical research, and other bioanalytical applications. So he is an expert in sample preparation and bioanalytical method development and has published several papers in this field. And he currently works in the clinical business unit in our Milford office. So without further ado, I'm going to turn it over to you, Jonathan.
Thank you, Rachel. And welcome to everybody who came today. So I'd like to start with sort of talking about what we'll go over today. One of the first things we'll talk about is a strategy. And you'll see this is maybe one of the most important parts, is having a systematic method of going through and developing a methods. We'll then talk about the sample matrices and analytes we have to deal with in the forensic toxicology world. I will talk about recovery and matrix effects, which is how we generally assess the quality of our extraction. And then we'll launch into sort of some of the options for sample extractions, techniques, products, protocols, things like that. How do we go about it? We'll then move into sort of the specifics of sample pre-treatment and extraction. What are some of the things that we have to consider? There are both matrix and analyte considerations, whether, depending if you're working with blood or urine or oral fluid, or if you have opiates or cannabinoids, that could all make a big difference. And then also assay requirements. How low do we have to go? Are we doing qualitative or quantitative work? What's our dynamic range? That sort of a thing. And then finally, we'll talk about a few application examples that you can see here. We'll sort of demonstrate some of these method development and optimization techniques. So before we get started, just to get a feel for the audience, we have our first poll question, and that is, what sort of sample prep are you currently using in your lab? It could be one, all of them, none of them. So if you want, just take a few seconds and answer that, and then we will move along. All right, I see about half of you have answered. We'll give it a few more seconds and we'll take a look at that. All right, we're over 50%. So it looks like we have a good spread. Some people doing dilution, protein precipitation, liquid-liquid, maybe more doing solid phase, and a few others. So we will move along. So the first thing I want to talk about is a strategy. You know, when we have a new method or we have to optimize an old one, and we don't just start doing things. One of the important things is our initial research. You know, I'm developing methods. You know, what is the analyte that I'm looking at? You know, what's its pKa? How will it behave under different pH situations? Is it polar? Is it non-polar? Is it stable? Do I have to worry about that? Is it generally protein bound, especially in whole blood or plasma? If it's in urine, is it subject to secondary conjugation? The other thing is, what's my matrix? There are differences between plasma, urine, whole blood, hair, oral fluid, etc. You know, each one of these has its own unique challenges to overcome. And this will really lead to our initial strategy. So we don't want to, hopefully we'll start with some knowledge like, okay, I have a moderately polar compound, it's a base, I'm doing it in urine, it might be conjugated, and that'll say, I need to deconjugate it, get the pH right, and then extract it versus maybe something in blood where you would have to precipitate it to get rid of the blood cells and protein. The next thing we want to ask ourselves is, what's our goal? As I mentioned earlier, is this sort of qualitative or general screening? Do we need do we need very accurate quantification? How sensitive do we have to go? All those will lead us to developing a fit for purpose method. There's no sense in using the absolute cleanest and expensive and time-consuming technique if you're just concerned about generally, is it there, is it not? So that's another important thing. And then finally, we like to have a systematic strategy for optimization and troubleshooting. We like to do things one step at a time. Don't change everything. It's often iterative. We'll do one thing and then that'll lead us to another. And the aim really is to understand the quality of an extraction. Things like recovery or sample or extraction efficiency, matrix effect, as well as the amount or existence of residual contaminants. All of those things will help us really understand and continue to optimize and give ourselves the best method we can have. So let's take a quick minute and talk about some of the forensic bioanalytical matrices we're dealing with. So especially in forensics, one of the big ones is whole blood for either on-site DUI or post-mortem cases. And some of the characteristics, it's about 50% blood cells, mostly red cells, but some white cells and lymphocytes, platelets, etc. There are very high levels of protein and phospholipids, and that can affect For example, protein binding of some drugs. Phospholipids, if they're not removed, can cause column fouling, MS fouling, and also can influence some of the matrix effects in ion suppression. Let's see. Urine is another very, very common matrix. It does not have the proteins and the things that whole blood does. However, many of the drugs can be conjugated, either with glucuronides or sulfates, etc. We often have a high concentration of salts and other small polar molecules, such as creatinine and urea. And you can see this tells us about recent past exposure, and it's used in a wide variety of environments. The other big one is plasma or serum. Like whole blood, it has high levels of protein and phospholipids. However, because it doesn't have the cellular content, it's often easier to work with. If there's no protein binding, you may be able to get away with just diluting and pretreating rather than doing a protein crash or protein removal. Some of the other less common matrices, but certainly growing in popularity, are things like oral fluid. Or it's become increasingly used in driving under the influence testing. It is one of the issues is not necessarily with the oral fluid, which has some protein and cellular debris, but the stabilizers and the buffers that these samples are stored in contain dyes, stabilizers, and other things, and those can affect your sample prep. We also have some differential partitioning in oral fluid versus plasma, so it's something to take into account. And once again, these are used in driving under the influence, anti-doping, and routine tox. Hair is another increasingly popular matrix because of its ease of collection and the lack of privacy issues. However, unlike the other matrix, it's mostly protein and solid matrix. They often have a low concentration in sample mass. Again, like saliva, there is some differential partitioning, which needs to be considered during interpretation. The advantage is we get very long-term detection, often for months, and it's good for long-term screening, DUID, postmortem work, and some others. And then finally, for postmortem, obviously, we can use vitreous humor. Very similar to plasma, but there's fairly low protein content, and fairly straightforward to work with. Now let's talk about some of the forensic analytes and their properties, which are, as you will see, are kind of all over the place. Some of their usual suspects or the regular ones are things like opiates, amine stimulants, and benzodiazepines. You can see these varying degrees of polarity. Many of them are acid, or many of them are weak bases. Benzos contain acids and bases. Others, somewhat newer drugs in the past 10, 15 years are things like cathinones, synthetic cannabinoids as well, newer ones, and boom, 2CBs and other new pharmaceutical substances. There are over-the-counter drugs, natural cannabinoids, but in all of them, our sample and pre-treatment and extraction strategies are influenced by both the sample matrix and the analyte. So now what I want to do is talk a bit about recovery and matrix effects, but here we are. And so recovery is basically extraction efficiency. We're asking how successful was a solid phase extraction or any extraction method at isolating and recovering a compound of interest. If I put 10 nanograms in there, do I get 10 nanograms out, or do I get 8 nanograms out? And that's all we're talking about, the efficiency, how much do we get out? Now the matrix effect is really asking the question, how has the extraction method and chromatographic method removed and resolved matrix components that can cause ion suppression or enhancement? So we're looking at the effect of the residual matrix on the sample. Does it suppress the signal? Does it enhance the signal? Are those changes consistent or not? That's really what we're looking at when we look at matrix effects. So these are really, in my experience, are key tools for evaluating our sample preparation methods. Calibration curves may indicate a problem, you know, hey, we've got some inaccuracies, nonlinearities, inconsistencies, things like that, but often not its nature. So often to really understand what's going on, we have to go back and look at our recovery and our matrix effects, not only for their magnitude, but also their consistency. And quickly, how do we do these experiments? Well, we will analyze 3 sets of samples to do these. The first one on the left is an extracted sample. This is similar to any sample, QC or calibrator you have, but we'll put a known amount of drug or analyte in there. We'll do an extraction and analyze it. We also have a post-extracted spike sample. In this case, we just extract blank matrix through our sample prep procedure and then spike the standards directly into these files at the end. And in this case, we are calculating the mass balance, so we're adding exactly the same amount that we started with into the blank samples. And that really tells us how much is coming through versus being left behind. Our final solution on the right is our standard solution. And it's similar to the post-extracted spike sample in that, well, we're not extracting it, but we are once again adding the same amount of drug there. We're just adding it to basically a solvent standard versus an extracted blank sample. And by using these 3, we can calculate our recovery from the equation on the left, our matrix effects from the equation on the right, and that'll allow us to understand really what's going on with our method. All right, now I'm going to go over some sample extraction techniques. What are our options? What are our products that are available, and the protocols? Before that, though, we'll do our second poll question, and that's, what do you find most challenging about sample prep? You know, is it where do I start? Is it optimization? Is it evaluation? How can I tell if it's doing what I want it to do or not? Or is it something else? So I'll give you a few seconds to go ahead and answer those. All right, thanks for answering those. So looks like most of you know where to start, but for those of you who don't, hopefully get you oriented. But many of you are, again, interested in optimization as well as evaluation of the sample prep method. So that's very good, because hopefully that's what you'll get out of this seminar today. Okay, there we go. So there are a number of sample preparation options. This is certainly not all of them, but this I believe covers the main categories. And from left to right, basically we're increasing the complexity of the sample prep method, and we're also increasing the selectivity and often the sensitivity as well. The other thing is we may be increasing the costs. And the choice of technique is really based on sort of a balance of all this, you know. What's the sensitivity needed? What is the selectivity? What is our throughput, etc. Going left to right, we have, you know, the simplest things, say dilute and shoot or filtration. We're really not doing much other than diluting the sample, maybe adjusting the pH. One of the next ones, if we look at protein precipitation, it's fairly non-specific, but we're doing a bit of cleanup. That's very useful for blood or plasma, those sorts of things. You know, we get rid of some of the major proteins as well. Going a little more complex, a little more selective, we have liquid-liquid extraction or supported liquid extraction, where we're extracting the sample or the analytes into a non-miscible solvent from our aqueous phase, you know, urine, plasma, whole blood, etc. And after that, we have solid phase extraction. That can be either reverse phase or to get more specific, mixed mode, where we add an ion exchange component as well. And while we won't talk about it today, some of the more specific options are some of the MU affinity purifications. We're actually using antibodies that pull down our analyte of interest. That's used for many protein, often protein or peptide analytes where we really need high sensitivity, but often for small molecules as well. Once again, we're getting increasingly complex and increasingly selective, and we really want a fit for purpose method that fits what we're doing for the assay. To expand on this a little bit, here's a figure from some work we were doing a couple years ago, sort of evaluating, we're using a single molecule, the pharmaceutical apixaban, but we looked at various different sample PET strategies. On the left, we're going from simple to complex. We see most of them have a fairly decent recovery, although as we get to the mixed mode, the mixed mode ion exchangers, we see our recovery goes up and our matrix effects are really reduced. On the left-hand side, again, decent recovery, but some significant ion enhancement or suppression. On the right-hand side, we're looking at one specific indicator, especially for plasma and whole blood, of residual phospholipids. As I mentioned, those can dirty up your column or mass spec or cause ion suppression of certain analytes. And we see simple things like protein precipitation, even or reverse phase solid phase extractions still have some significant residual phospholipids. But when we get to more specific things like HLB prime, which adds phospholipid removal to reverse phase, or mixed mode ion extraction, we get cleaner and cleaner samples. So a couple different ways of looking, and really beyond the and sort of evaluating again the quality of our extraction. You can look at all these and say, okay, is it efficient? Am I getting ion suppression enhancement? And what are the residual contaminants left in my sample? Just a quick review of many of the sample extraction or sample preparation products available. These are obviously, this is products that Waters produces, but many other manufacturers make similar ones. There are things like precipitation or phospholipid removal plates. We have a Sirocco protein precipitation plate, which is just a filter plate, or we have OSPHO that allows us to remove the phospholipids and precipitate as well. There are things such as silica-based solid phase extraction products, such as in Waters, it's SEPAC, but there are others, again, other manufacturers make them as well. And finally, there are polymeric solid phase extraction products. These are probably used more frequently than any now. And those include things like Oasis HLB, Oasis HLB prime, which is reverse phase that adds a phospholipid removal, as well as our Oasis mixed mode sorbents, strong and weak cation exchangers, as well as the strong and weak anion exchangers. Just a quick diversion into HLB. We have presented this before, but it's an easy to use water wettable sorbent. It's really designed to have good balance and retention between polar and non-polar, reverse phase retention and polar retention. It's stable across a wide pH range, gives high recoveries for a variety of molecules, and it's water wettable. So it doesn't, unlike some of the previous sorbents, it does not dry out under vacuum or positive pressure. There are a couple of reverse phase sorbents I mentioned. Oasis HLB is simply the copolymer alone, and Prime HLB is a copolymer with the addition of a phospholipid removal sorbent as well. There are also numerous mixed mode sorbents. As I mentioned, the weak and strong cation exchangers, and then the weak and strong anion exchangers, as well as Oasis Prime MCX, which combines strong cation exchange with phospholipid removal as well. Here's a look at the structure of the Oasis sorbents. You can see in the middle, the Oasis HLB shows you the hydrophilic monomer as well as the lipophilic one for reverse phase retention. And onto this backbone is added one of the four ion exchange functionalities that you can see here. One thing to be very aware of when doing ion exchanges, to be aware of the pKa of both the ion exchanger and the analyte as well, and the ion exchange capacity. Often if you're using, say, a urine sample that's highly buffered, you may need to use a larger mass of salt sorbent to prevent that from being overwhelmed. Now that we've looked at those, let's talk about some SPE protocols. So this slide shows us a very generic solid phase extraction protocol where there is a sample pretreatment phase, and that may be precipitation, enzymatic deconjugation, or simple dilution and pH adjustment. With the silica sorbents, we used to have to condition and equilibrate them. However, with some of the new, certainly the Oasis sorbents and other polymeric sorbent, that is not often no longer necessary. So we can skip it. Nevertheless, after our pretreatment, we're going to load our sample. Then there'll be one or two, possibly more washes to sort of clean it up and get rid of any contaminants we want to eliminate. And then finally, an elution, usually in a high organic solvent, and then we can either evaporate, reconstitute it, dilute the sample, or simply inject it directly, depending on the assay. If we look at Oasis HLB and Oasis Prime HLB, your standard reverse phase sorbents, these are two starting protocols. And we're going to be talking about optimization, so clearly this is where we begin. But you see they're very, initially they're very straightforward. We're going to load our treated sample, then we're going to wash it. Our starting protocol with reverse phase is usually low, 5% methanol, low organic, to make sure we're not washing our compound off the sorbent, and then an elution and high organic. In the case of HLB, the standard is the starting point is methanol. For prime HLB, it's acetonitrile methanol. So I mentioned that we may want to modify the wash or the elution, and this is the sort of thing we're looking to do. Whenever we're doing an optimization strategy, we want to sort of look at, not just a single, not just a single condition at a time, but maybe multiple conditions at a time. Not pH and organic content and all that, but change one thing over time systematically. And here we see an elution profile for a generic molecule. And what we see is when we are, assuming this is a reverse phase sorbent, when we're washing with low percentages of methanol, we have no peak area. Basically, everything is staying bound to the sorbent. However, the higher the percentage of organic we get to a point of, say 70, 80%, where we're eluting all of the molecule off, or all of the analyte off the sorbent. And we want to, and so this is what we want to know. We see here in this figure, now doing this experiment, I can see where my optimum wash is. So I don't want to wash more than that, or I'll pull it off, as well as where optimum elution. If I elute with a weaker solvent, I'm not going to get it all off. I'll have lower efficiency. To show an example of this, here's an experiment we did a few years ago where we're looking at optimizing washes for a number of benzodiazepines on Oasis HLB. And as you can see, our wash step consists of 20, 40, 60, or 80% methanol. And as we can see, if we look at these for 20%, 40%, we got good recovery for all of our analytes. However, when we started increasing, above 50%, 60% methanol, 80% methanol, we're starting to lose recovery for many of these compounds. And that's because that wash is pulling it off, is rinsing it off the sorbent into basically our waste container. So by looking at this information, you can say, okay, to get all these efficiently extract all these compounds, I'm going to stay below, 40% methanol wash. And this is a sort of systemic strategy that we want to go through when we're doing optimizing any part of the method. So our final poll question is more general: is what are your primary concerns with sample preparation? I guess again, are you concerned? Does it have to be easy to use? Is cost a big consideration? Do you want it to be reliable so you can depend on it? Or is it the absolute cleanliness the most important thing? So I'll give you again a few seconds to answer this question, and then we'll continue on. All right. So it seems, thanks for participating. It seems for many of you, reliability is the most important thing. Not terribly surprising. I'll be happy to tell all my sales folks that you guys aren't terribly concerned about costs. I'll be very happy to hear that. Again, ease of use. A little surprised because often, we have people struggling with that. But again, thanks for participating. It's useful information for us. So now I want to go through a couple specific protocols, and these are what we call at Waters. These are the Oasis 2x4 protocol that we use for our mixed mode sorbents. And this is really a strategy for quickly designing an ion exchange or mixed mode ion exchange method for forensic and bioanalytical methods. So just to remind you, on the left, we have the Oasis family of sorbents, including HLB, as well as the other ion exchangers. And on the right, we have the 2x4 method development protocol. If we look a little closer at that, it looks complicated, but really we're looking at basically two protocols. On the left, we have a protocol for bases and strong acids on the complementary sorbents, where we load our pre-treated sample. We wash under acidic conditions. We then do a second wash with 100% methanol or 100% organic. And that's generally designed to get rid of all of the neutral interference, as many of the neutral interfering substances as we can, while retaining our compounds by ion exchange. And then finally, we're eluting in high organic and in alkaline conditions with 5% ammonia and methanol. The second protocol, again, we're loading a sample. In this case, we're washing with an alkaline solution. Again, for the same reason, we want to get everything, all the charged species in the correct state for a full ion exchange binding. The second wash is the same, 100% methanol. And then we do an elution, again, in a high organic. In this case, we're using acidic solution, 2% formic acid, to turn off those ion exchangers and allow the reverse phase to take over so we can elute the compounds. Very quickly, I want to take a look at a couple of retention maps. You may notice these look similar to the reverse phase retention map we were looking for earlier, but instead of being based on organic content, this is based on pH. So this one is for the Oasis MCX. It's the strong cation exchanger. And what that means is that the sorbent is always charged. It's at a permanent negative charge, so it can attract positively charged bases. So as in the one protocol, when we load it under acidic conditions, our base will be ionized and it will bond by ion exchange to the sorbent. And as you can see, it's locked on there. We can't get it off unless we change the pH. When we do that, however, you can see to the right of the figure, that lock turns off. We have poor retention, and it's easy to get it off by eluting with 100% methanol or some other organic. One thing you notice is that in the middle, there's a very steep section of this pH curve. And one of the things that's very important when doing method development is we want to stay at least 2 pH units away from the pKa of our analyte and our sorbent as well. So if we have a wash or a dilution around, somewhere, 6.5 to 9.5, we're going to be that steep section. We're not going to get fully efficient extraction, or we're not going to get good retention. We're going to get some breakthrough and leaking, and we're going to have some issues. So in this case, we wanted to say, we would want to stay to keep things bound lower than, say, pH 4 or 5, and to elute them nothing lower than pH 11 or 12. We see a similar curve for the weak cation exchanger. The difference here is that instead of changing the ionization state of our compound, we're changing the ionization state of the sorbent. Once again, here we see a strong base, it's always ionized, and by changing the pH, we change the charge state of the sorbent, allowing for either strong retention or easy elution. So just a quick review of Oasis solid phase extraction sorbents. We've got HLB, a copolymer sorbent with polar and hydrophobic retention properties. It's water wettable and gives good results for a wide variety of compounds. We have Oasis Prime HLB that's designed to enable simple, clean, and fast protocols and adds phospholipid removal to the mixture. And then finally, we have the Oasis mixed mode sorbents and the 2x4 method. And that really gives us a simple approach to mixed mode SPE development, and is really used, is what we'll use when we want sort of the cleanest and most specific sample prep we can get. Once again, understanding ion exchange retention curves is critical to using these sorbents. Okay, now I'd like to get into some of the details, and we'll start by sample pre-treatment and extraction, some of the matrix and analyte considerations, as well as some optimization examples and strategies. So urine, let's start with that. As mentioned before, it tells us about recent exposure. Often pH adjustment may be sufficient. However, many drugs can be conjugated, and as I mentioned, have a high concentration of salts and small polar molecules. So some of the things we'll want to do is deconjugate the glucuronide metabolites using chemical or enzymatic hydrolysis, and obviously adjust the pH before and after hydrolysis. There are a number of beta-glucuronidase types. Some of the older ones were derived from mollusk species or bacteria. More and more people are going towards a recombinant engineered glucuronidase enzymes. These are made by companies such as Cura and IMCS. We're not endorsing either one of them, but they are very popular, and tend to give very good, quick, and efficient hydrolysis. But one thing to remember, regardless of which type you're using, is all of them have optimum pHs, temperature, and times, and those can be found often in the manufacturer's instructions or information. Here's just a quick example of a hydrolysis optimization we did in our lab a few years ago. And we were looking at, basically, we took some conjugated opiates and benzodiazepines, or morphine, oxymorphone, lorazepam, etc. And basically we incubated them in our hydrolysis enzyme, and then basically monitored their conversion from the conjugated to the free state. And as you can see over here, we did it from 10 minutes to an hour. Many of the benzos especially were immediately hydrolyzed. However, some of the opiates, it took a bit of time. In this case, it really needed a full 30, for oxymorphone and morphine 6, full 30 or 60 minutes. As I mentioned, many of the modern enzymes are a little quicker, but it's always good to take a look in your own lab to optimize it and make sure that you're getting full deconjugation. Some of the issues we can run into if we don't fully deconjugate are basically underestimating of the drugs that we've got in our sample. Here's a paper or a figure from a poster that was presented by one of our colleagues, Dr. Tarmazzano, at the Society of Forensic Toxicology this fall. It'll be published in a paper later on, but just showing the degree of conjugation of a number of forensic analytes. I'm not going to go through all of them there, but they're, as you can see, they're in this figure, they're ordered from the least conjugated to the most conjugated. So things like, pregabalin and hydrocodone on the left, if you can read that, are almost all in their free state. However, other things, other compounds, such as some of the benzos or cannabinoids, are very highly conjugated. And if this is not addressed, we can miss them or underestimate their presence in a sample. Now we'll get into whole blood and plasma pretreatment. They're going to be fairly similar. We've talked about the properties before. Usually we are, for whole blood, we're going to precipitate it, often with, sometimes with a salt such as zinc sulfate, but often with methanol or acetonitrile to disrupt the protein binding and clean it up. We can also do both. We'll then center and remove the supernatant and dilute it with an aqueous to reduce the total amount of organic to under 20%. This helps prevent breakthroughs of analytes, and at that point we can also include modifiers as appropriate for the extraction. For plasma, fairly similar. However, because we don't have the cells, if things are not highly protein bound, we can often just dilute it as a pretreatment. However, if drugs are protein bound, we'll want to do some again add something like zinc sulfate or precipitate with methanol or acetonitrile to disrupt that binding. What are some of the consequences if we don't do this properly? Well, here we see a couple of figures where we're extracting steroids from plasma. On the left, we've just used a simple pretreatment with phosphoric acid. On the right, we've actually treated with methanol and zinc sulfate. And you can see cortisol and 17-hydroxyprogesterone are well recovered, but androstenedione really requires that protein disruption from from that combination to get it out of solution and get an efficient extraction. Otherwise, we're only pulling out about 10% of it. Another quick example, looking at coenzyme Q10, we've plotted sort of the recovery with the ratio of plasma to solvent use. This is a very, very hydrophobic molecule we worked with a few years ago. And in this case, we needed to actually go up to a 6-to-1 ratio. So 600 microliters of solvent to 100 microliters of plasma. This is a fairly extreme example, but again, it gives you a good example of how systematically evaluating this allows us to easily pick where we want to be. Now, oral fluid in the hair samples, they can be a little different. For oral fluid, we're often going to dilute and adjust the pH, add acid, base, or a buffer to adjust the analyte sorbent charge. Often there's not protein binding. We don't have to worry about that, but we will want to evaluate the recovery from the device to make sure we're not getting too much absorption on the pad. For hair, it's a little different because it's a solid sample versus a liquid like the other ones. That will involve washing away surface contamination, things like homogenizing the sample, digestion or extraction to release the compounds, extraction with an organic solvent, and then even maybe a solid phase extraction after that. So hair can be, because of its nature, can be very complex. But again, you see a lot of pre-treatment steps to consider. Okay, now that we've talked about a lot of these, I want to get into some of these application examples, and these will include some optimization and examples of how we either altered or optimized the pretreatment or the extraction, the washes, etc. to get the best performance out of our assay. The first one we will look at is a definitive drug testing method for urine. In this case, we use a Oasis mixed mode strong cation exchanger, Oasis MCX. And we were looking at a number of drugs, including opiates, benzos, amine stimulants, some of the new cathinones, et cetera. So a wide variety of drugs. Looking at the pre-treatment, we see we are hydrolyzing it using a hydrolysis buffer and beta-glucuron ice. That goes for an hour. This was done a few years ago. And then we simply, after the hydrolysis, we pre-treat it with phosphoric acid to get the pH to the state that we want. The extraction is fairly straightforward. We load the sample on the MCX microelution plate. We have a single wash. In this case, instead of 100% methanol, as the original protocol calls for, we use 20% of methanol in water. This is because of some of the benzodiazepines are just bound by a reverse phase, and we want to make sure we don't wash them off. And then the elution is fairly straightforward, organic 50-50 acetonitrile methanol with ammonia, and then this is simply diluted with mostly water and a little formic acid for injection onto the system. Going into a little more detail, as I mentioned, sort of the mixed mode strong cation exchanger, the bases are bound via ion exchange. However, some of the benzodiazepines are acidic, and they are bound by reverse phase. So, as I mentioned, we back off on the wash and use both the ion exchange and the reverse phase sorbents to cleanly extract our sample compounds. Looking at the results, we see very, very good efficiency of extraction all across the whole samples. There are a couple of them that don't quite behave, but the vast majority of them had very efficient and reproducible extraction. Another example is looking at fentanyl and some of its newer analogs in whole blood. This is work we did a couple years ago, published as an application note as well as a peer-reviewed paper. So in this case, we have a couple things. We're looking at whole blood, and so it's a little more complex. So we start with our sample pretreatment. In this case, it's a 1-to-1 with zinc sulfate and ammonium acetate, followed by a second 1-to-1 precipitation with acetonitrile and methanol. We then spin that and then dilute it with supernatant for the solid phase extraction. You'll notice in this case, we're just using a 1-to-1 precipitation. We don't worry about protein binding with fentanyl and these other its related compounds, but we do want to clean up the sample. After the pretreatment, we used Oasis Prime MCX. We loaded the sample. As you can see, our wash is a little different. We had the protocol for this is to use ammonium formate with formic acid. This allows us to wash away the residual phospholipids in the sample as well. With that second wash, again, the elution is straightforward, ammonia with acetonitrile methanol, and then the similar dilution. Looking at some of these results, if we first sort of look at our quality of the cleanliness, we look at our extraction on the bottom with Oasis Prime versus a simple precipitation, and then graph those phospholipid areas. We see we're getting rid of about 96% of the phospholipids. So very, a clean extraction that'll help our method. Looking at the quality, our recovery and matrix effects, we see excellent recovery. 60, mostly around 80%, with low standard deviations. And in this case, we almost had did a double take because our matrix effects were eliminated for all of them. We just didn't have to deal with it. And if you're lucky, sometimes this is what you can get with a nice clean extraction. We don't have residual things to mess up our assay or foul our instrument. We get good, clean data. And the final example I want to use is sort of THC and its metabolites in whole blood. Again, we're going back to whole blood instead of polar compounds. These are sort of non-polar, more hydrophobic. They tend to be highly protein bound as well. So in this case, we were adding a little bit of zinc sulfate and then precipitating with 3-to-1 acetonitrile containing a little formic acid. We then dilute it with water, and our solid phase extraction is, again, straightforward. We load the entire sample. We wash with 25% methanol that, just like earlier with the benzos, has been optimized for these analytes. And then our elution is, again, is just a 90-10 acetonitrile methanol, and then that sample is diluted with water. If we look at our recovery and matrix effects, we see the sample gives us excellent recovery at greater than 80%, as well as minimal matrix effects. We don't have big ion suppression to worry about. So we get a nice consistent data from that. So to get now a little more detailed or a little more specific, I want to talk about some I guess fairly customer specific optimizations we did for looking at THC and related compounds in oral fluid. So one of the things we had to look at was extraction from the device. We ended up adding a mil of acetonitrile to our collection device and letting it sit there overnight to make sure we didn't get absorption on the pad. We'll see a figure on that later. And then we also modified our SPE method with this a wash containing 5% ammonia to give us a cleaner sample as well. Taking a look at this, the first part, we found we weren't getting the efficiency or the extraction recovery we expected from the device, from the whole method. But adding a little bit of acetonitrile in this case really improved our recovery by 20 or 30%. And so we think what was happening there is we were getting actually absorption of these compounds onto this this polymeric pad that's used for collection. Again, looking at this modified method, excellent recoveries, very low matrix effects, give us a nice clean method we can rely on. So we also did some extraction optimization for this one as well, in addition to the pre-treatment. Here we see some method optimization where we are looking at different types of washes. I'm sorry, not different washes. Different type of washes, where we are doing it with sort of different combinations of water and organic. We've added acid, we've added base, and we found that of all of these, the one in the middle, this 5%, this wash with 5% ammonia and 5% methanol really helped clean up the sample. For whatever reason, it gave us minimal matrix effects. And so that's the one we went with for the final method. We also did the same thing for the elution. Once again, this is during sort of the method development. We're looking at, acetonitrile water or IPA or different ratios. And again, on the far left, we see that when we use 90% acetonitrile and 10% methanol, once again, very, very low matrix effects. So these are some of the optimizations you can do after you get the basic method settled out. So in conclusion, I just want to re-emphasize that both the matrix and analyte both need to be considered in our sample preparation method development. Do yourself some research and save yourself some time and struggles. So, understanding the nature of the solvent, is it conjugated, is it protein bound, what's its pKa, can go a long way to help you choosing the appropriate sorbent and appropriate conditions as well. We can use our recovery and matrix effects to fully understand the quality of our extractions. And again, we want to use a systematic strategy for optimization. Change things one at a time to fully understand, where you need to be and where you need to go to get the best extraction possible. Here are some of the references of some of the figures used in this article, if you care to take a look at them. And then finally, we'd be happy to take any questions from the audience. Oh, and here's our contact information if you're interested in contacting us directly. Thanks.
Thank you, Jonathan. We do have a couple of questions here in the chat. So if you have anything for Jonathan at this time, please feel free to ask your questions. We've got a few minutes left here to do some Q&A. So the first one that I have here is when evaluating extraction performance, what is a good rule of thumb acceptance criteria or percentage range for recovery and extraction efficiency?
I'm sorry, could you repeat that again?
Yeah. No problem. So when evaluating extraction performance, what is a good rule of thumb acceptance criteria or percentage range for like recovery and extraction efficiency? What would you typically want to look for in that?
So recovery, you want it to be as high as possible. Obviously, we would ideally like to see things at 70, 80%. However, the most important thing is probably consistency. I don't think in bioanalytical there's a cutoff, but if, and I've done, sort of certain assays where sometimes all you can get is 20%. That's not ideal, but I would say, a goal should be at least 50%, but more important, you want to get, 15, 20, at least at the most 15 or 20% RSD, because your consistency is really going to be critical, and especially your accuracy, if that helps.
Yeah, absolutely. And then we have another question here. It says, thank you very much for the presentation. Would there be any subtle difference in sample prep serum versus plasma?
Usually not. Serum is going to be a little bit cleaner. It may have a little bit fewer proteins, but I think you'll still get protein binding. You still have a lot of phospholipids. So generally what works on serum should work on plasma as well. I think the one issue is really going to be, it's a drug, do you need serum or plasma specifically for the drug itself? How does it behave? But the extraction techniques should be pretty much the same.
Okay. And kind of talking a little bit about the extraction technique in your mind, I'm not sure if this is something you covered, but wanted to go over it anyway. What would you consider is the cleanest sorbent to use?
The cleanest is usually going to be the most specific. So for example, I would say a mixed mode ion exchanger, because you can retain things obviously by reverse phase. We can also retain things by ion exchange. We can optimize the washes a little better to get rid of interferences. So that's going to be your best sort of standard sorbent, unless you go to things like immunoprecipitation, where you're specifically looking for one single molecule.
Okay, great. And then, so this question, I'm not sure if we have an answer now, but we can always look into this, do our application notes have the CE-IVD certification?
I believe no. These are, what we're talking about today is all forensic. So it's, it's not related to IVD. Right. The only IVD product we have are the TACRO, immunosuppressant monitoring, and that's a whole different kettle of fish. But no, these are not IVD certified. These are just examples, good examples, but examples.
And research based, yep, absolutely. Are there any additional questions that we have? We're kind of running up on time. We've got about a minute or so left. We'll give it a section, but while we wait to see if there are any other questions that come in, I do want to put in a little plug about the fact we are holding a second webinar on July 22nd, same time. And this particular webinar will be on LC troubleshooting. So kind of following up on this, we talked a lot about the sample prep. The next one will be on the LC troubleshooting. So please be sure to sign up for that and get a chance to fill out the survey when you do get a chance. We do have one last question that came in that we can probably sneak in here. So
sure.
This is from Destiny. So I'm currently working on a urine drug screen method. We're testing out using cold acetonitrile and then filtering the sample with syringe filters. Everything is extracting, but a handful of analytes, the opioids in particular, are not meeting qualitative acceptance criteria due to looking to be dirty. Would it be worth assessing if the filtering step is working by injecting a filtered sample versus an unfiltered sample, since they're seeing so much matrix components in their reagent blank?
Let's see. I'm reading the question again. Just it's a long
Okay.
question. So,
It's kind of trying to decide about, and this is also something we could talk about offline too, and reach back out to discuss further.
Yeah. Again, feel free, as Rachel said, feel free to reach out offline, but quickly, I would say, yes, it's, it couldn't hurt to look at, not to look at the filtered versus the non-filtered. You wouldn't, at the very least, you get some idea of cleanliness, but you also tell maybe if some of your compounds are being held up somewhere. But that's probably a good idea. But again, what do you reach out, and specifically, it might be easier to give a more detailed answer offline.
Okay, awesome. Well, with that, I believe we have reached our hour. We're a little bit past. So again, I want to thank everyone for joining today. And you will receive a thank you email with some additional information and a copy of these slides for your reference. And we look forward to seeing you at a webinar in the future. And thank you, Jonathan.